the Cryo-3, which it doesn't do for the other two.
Okay.
But either way, we're good to go.
Okay. All right.
Yeah.
Welcome to the Simon's Electron Microscopy EM winter course. Today's lecturer is Dr. Oliver Clark. Ollie obtained his PhD at the University of Melbourne, and he is currently an assistant professor at Columbia University, where he's using Cryo-EM to study molecular machines like the ryanodine receptor that control intracellular calcium signaling. Today's lecture is titled Tips and Tricks for Manual Model Building of Atomic Models into Cryo-EM Maps. And basically what that means is what you do with that medium to high resolution Cryo-EM map, how you build an atomic model into that to tell a biological story of what your research is trying to elude. So without further ado, I'm going to hand the stage over to Ollie.
All right, thanks very much, Laura. So this is going to be pretty informal. I just want to give you guys a guide to what to do when you start with basically a map and a sequence, and you want to figure out how to generate the best possible model that fits your map, your density. So the way I like to think about model building is that it's basically a way of creating the most compact representation of both the information that's encoded in your density map and all the prior knowledge that you have about your protein. Both prior knowledge that is general, so I'm thinking protein secondary structure, protein chemistry, the interactions between amino acids, hydrogen bonding interactions, and interaction that is specific to your protein. So for example, like the particular secondary structure prediction that you can obtain for your protein sequence, predicted disorder properties, known folds that you can predict from sequence, things of that nature. And so at medium resolution, so in the sort of 3 to 5 angstroms range, which is still, what we're most commonly dealing with, I would say, this still a lot of the time requires manual model building. So placing folds by hand de novo and figuring out how everything fits together. There are automated building tools for Cryo-EM maps. Probably the two that I would say that are currently best are the tools in Phoenix, so Phoenix Map to Model, and MainMast, which I haven't actually tried, but the results look very promising. However, in those cases, even with a relatively high resolution map, you're still going to end up with a fairly incomplete model in most cases, and it's still going to require a lot of manual interpretation in order to fit all the fragments that you get out of auto building together, decide which ones are right and which ones are wrong. Basically, you still need a person in the loop to judge whether the machine has done the right thing, and I think that'll be the case for quite some time. And so in this process, the major trade-off is really between between the prior knowledge that you have about your protein and what information is encoded in the density map. And what I mean by that is that in the extreme case, if you have a 10 angstrom map, but you have perfect knowledge of the protein structure that you want to fit to it, that's easy. That's trivial, generally speaking. And likewise, if you have a 1.5 or let's say 1.3 angstrom map and no knowledge of the protein sequence, well, actually even there are ambiguities, but you would almost be able to fit your structure without any prior knowledge whatsoever. But usually we're dealing with something in the middle, and you need to figure out to what extent you can build a model that is a unique model that fits both the density and your prior knowledge. Okay, so as I mentioned, some examples of prior knowledge, things that you get from your sequence. So secondary structure predictions, disorder prediction, information that you can obtain from sequence alignment. So I'm thinking particularly about evolutionary couplings, which you can get from analysis of many hundreds of sequences, looking at how substitutions at widely separated pairs of residues are correlated. The idea being that if you have, let's say, an arginine in position 1 and a glutamate in a position 100, and whenever they're substituted, those two positions swapped around, that's indicative that there might be like a salt bridge between those residues, and you can create distance-based restraints based on that. And of course, just like patterns in the sequence, patterns of large and small residues, and knowledge that you have of the folds of similar sequences that you can find from looking at bits of your sequence. From the map, things that can inform your model building, obviously the resolution controls what level of features you have. So roughly at like, let's say 12 to 9 angstroms, you'll start seeing alpha helices. Anything lower resolution than about 12 angstroms, you can see overall shapes of domains, but not secondary structure information. At about around 4.5 angstroms, you'll start to see separation of beta strands, and that's another critical point. And then at sub 4, you will start to see well-defined side chains of amino acids where you can start to distinguish bulky amino acids. And once you get to 3, you can really start to distinguish, sub 3.5, let's say, you can really start to distinguish individual side chains from one another. Let's say like a tryptophan from a tyrosine from a phenylalanine, for example. So we want to take all of that information and combine it to get a unique model that agrees with both sets of information. And if you can't do that, then you should specify that ambiguity. So don't build sequence where you can't justify sequence placement. Like leave residues as unknown if there is ambiguity about the sequencing, and that's totally fine. Anything that you can and should do if you're not confident of the sequence register in a particular region of a protein, because it's more informative for people to know which, because a lot of people will only look at your model, and so it's more informative for people to know like which regions you're not certain of when you're building. And yeah, we'll go through all of this. Okay, so I think before you get anywhere near like looking at your map, what you want to figure out is what information can you obtain just looking at your protein sequence, just analyzing your protein sequence. How much can you know about the domain architecture of your protein and the secondary structure and all of that stuff, because that's very, very informative when you come to try to build your first model into a map. This is the wrong slide for that, I'm sorry. Let me just come back to that. But yeah, okay, I'll come back to this in a minute, but basically, before you start with a map, make sure that you have all the maps that you want for building. I would usually suggest building with multiple maps, so either convert your map to an MTZ file and then adjust the sharpening on the fly, or just have several maps sharpened at different levels so that you can, because there's a trade-off between the level of sharpening and detail and connectivity. Less sharpened, you'll have better connectivity, but worse definition of high-resolution features like sidechains, and more sharpened the converse. And I would try different methods for sharpening your map. I quite like Phoenix Auto Sharpen and the method that's used in System, which is based on spectral widening as opposed to just simple negative B factor range. And the other point I wanted to make is if your map doesn't look like the nominal resolution, trust your eyes over the FSC. There are many things that can, give you a nominal resolution that is not consistent with the features in your map. The features in your map are what matter. So for example, if you have preferred orientations of your particles, you can have a map that will look very stretched and it will be nominally a very high resolution in one direction but poor in the other direction. And I would always analyze that using a 3D FSC approach. So there's a server from Dmitry Lumkus at Salk, which is great for that. And also analyze your local resolution variation, so you know what level of information you have in different regions of the map. And double check your mask that you're using. You should always know what mask you're using for FSC calculation as well. All right. Yeah, now, as I was saying, in preparing for model building, you want to figure out what you can learn from the sequence alone. So there are a few things here. I'm only going to cover a couple of them in detail. But this is my general workflow if I'm starting a new model building project. So first off, I'll start by trying to identify what conserved domains I have in my protein and where are they. And generally, I'll do this using the conserved domain database from NCBI, which you can also access using Delta Blast. If you search your sequence using Delta Blast, it will come up we'll try to identify any conserved domains for which there are already structures solved. And that's really helpful when you then want to go to the next step in this workflow. So then you want to get initial structural templates to fit to your map. There are a whole bunch of approaches you can use for this. I really like Fugue. There's actually one that I didn't mention here. I think it... Right, there's another one called Muster from the Zhang lab, which is really good for finding threaded models of your individual domains. Yep. Okay. Then the next level down is you want to get to understanding your predicted secondary structure. I generally do that using Extelpred, which was designed to predict protein crystallizability, but also just generates a really nice compact summary of all the sequence-derived heuristics. So secondary structure from JPred, predicted disorder from DisoPred, and transmembrane helix prediction as well. And then you can add on top of that like contact prediction from evolutionary couplings. And I would always also perform an analysis of sequence conservation. So make a really nice multiple sequence alignment of quite a few diverse orthologs of your protein of interest so that you can identify regions where there's variation, regions that are highly conserved, because the regions where you have gaps or insertions are generally in loops, and it can be helpful when you're building. And I would also advise to, annotate and update that as you go. So annotate it with secondary structural information that you derive when you're model building as you go along, because it'll be and any mutations and anything, so that you've got essentially like a blueprint for your protein that'll be then really useful when you're analyzing your structure. Yeah, so as I mentioned, so this is what I was referring to. Whoa, this is one slide out of sync. Sorry. There we go. Yeah, so this is sort of the interface. This is what I was referring to. That doesn't work. This is what I was referring to regarding the conserved domain database. So this is for the ryanodine receptor, which is a very large multi-domain protein. And you can see that you can identify a ton of different conserved domains, like just from the get-go, which are then really useful if you have a map that you want to start modeling, because they provide anchor points that you can use both to model those parts, but also to give you sequence anchors for the regions that you don't know, for the regions for which there are no sequence identifiable structural templates in those adjacent regions. And if you click on any of these, you can expand it out. It'll give you like a sequence alignment. You can download structures, and it's very, very useful. I would recommend having a look at it. But you can see that not all of the sequence does have structural templates. And once you have an initial model trace for one of these regions, I would suggest kind of doing another round of this. So taking your initial C-alpha trace or polyalanine trace of a new domain that you've just built at a main chain level, and then plug that back into a server which will take coordinates and search for folds based on structure. The classic one is DALI, but you can also use PDB fold. There are probably some other ones as well, but those are the two that I'm familiar with. All right, so secondary structure prediction. Yeah, so I mentioned extilpred. So this is an example of sort of the output you'll get from extilpred. It gives you just a general compact summary of everything with numbering, where red is alpha helix, blue is beta sheet, and any region that's underlined is predicted to be disordered. The reason why this is really useful is that secondary structural prediction is actually extremely accurate. So it's a very strong constraint on your model building. So JPRED, I think, is like 82% accurate, and some of the newer neural network models, like Spider-3, for example, more like 85 or maybe even higher percent accurate. So what that means is if you encounter something that in the sequence is strongly predicted to be an alpha helix, but you're not seeing alpha helical density in the map, it's an indication that you probably built it wrong, and that you should maybe reevaluate whether your sequence register is correct in that case. And you want to have pretty strong evidence that it is if it's conflicting on a broad scale with secondary structural prediction. Like, disorder is not quite as strong. Like, some regions that are predicted disordered will actually be ordered, but it's still a good thing to pay attention to so that it can give you additional confidence. If you run out of density and you're also hitting a region that's predicted to be disordered in the sequence, that gives you extra confidence that you got it right, that you're okay so far anyway. All right, so a specific example of that, like if you take a look at this region of secondary structural prediction, and this is what you see in the map, like you should be able to say almost, within a few seconds where the first candidate location that you're going to choose would be, right? Because you can see that almost everything here is predicted alpha helical. But there's one location, maybe a couple of other candidate locations, but really just one location where you have a very strongly predicted pair of beta strands. And so that would be the first region you would choose to anchor yourself on. And, yeah, so basically that's what I was describing. So that's all describing like what you can learn from the sequence. If we just put all that aside for a second and think about like what you can learn just from the map. So first off, there's something wrong with this picture, which you may be able to appreciate. Can anyone see what's wrong with this?
It's a left-handed up.
Yeah, it's left-handed. And that's really obvious in this case because we're at relatively high resolution. But like if you're at 4.5 angstroms, that can be tricky. And the reason why this is even a consideration is because in Cryo-EM, you can converge, if you're solving a structure ab initio, you can converge on the right-handed or the left-handed, like the correct or the inverted model with equal probability. So it's something that if you're working particularly in the lower resolution range, like the 4.5, 5 angstrom range, you want to be paying attention to that. And obviously, like if you're fitting a solved domain, always try fitting it into both hands of the map, because that's a very quick way to determine that. Yeah, so other things that we can learn here. So the first thing that you want to figure out is which direction, like if you're thinking about helices, which direction does your helix point? The sort of trick for remembering this is that helices are more or less like Christmas trees, where the N terminus is at the base of the Christmas tree and the C terminus is at the top. So all your side chains point down towards the N terminus of the helix. So from that, you should be able to tell more or less which direction this helix is pointing. So we'll try that one. And then we want to figure out, looking at this, like whether we can determine any probable side chains from the density. So there might not be anything you can say with absolute certainty, but like you can see this one up here near the end, like that's obviously like a large aromatic. So that would be what you would start with first, and there's another relatively sizable residue over here. So you might try like a tyrosine, a phenylalanine, a tryptophan, and then see how that goes. So try phenylalanine, try a tyrosine, try a tryptophan. It actually looks most like a tryptophan in this case. And then what you'll want to do is to test your initial hypothesis, usually with several of those anchor residues, by extending that sequence assignment in both directions along the chain. And if you got it right, it'll make sense. And if you didn't, it won't, or it'll break, or that hypothesis will break down at some point. And it can break down because you got it wrong, or it can break down because you ran into a region where you thought it was an alpha helix, but actually there was one extra residue in that turn, or things that take you out of sequence register. Let's say. Right. What was I saying here? Oh yeah, the other thing that I wanted to point out here, like you get a lot of information from large aromatics, but actually in a way you get even more information from very small residues, in the sense that if you have a residue which you've assigned as a glycine, but you see a density for that residue, you know it's wrong. Like you know 100% that that's wrong. So the combination of patterns of large aromatic residues and really small residues like glycine, alanine, proline, serine is really helpful. And you want to be looking to begin with, if you're just starting without a model, like without any homology models or anything, you want to be scanning your density and your sequence to find regions that are information rich, that have unique patterns of large and small residues, which can help to anchor you when you're building. Which is more, so this is an example, right? So if you had a sequence which was like VT, VVVV, all of those look more or less the same at low resolution. And so you can't tell that. Whereas if you have something that's like FGAY, like you know already that even at 4 angstroms, you're going to be able to see that and hopefully uniquely identify it. And if you get enough of these and you can tie them together, that's how you are able to build a complete structure.
How about these negatively charged side chains?
That's a good point. Yeah.
They are very hard to detect even at 3.1. I have 3.1 angstrom now, so they are hard to detect.
Absolutely. So they get wiped out by radiation damage. And that's definitely something you should be aware of when you're building in Cryo-EM versus crystallography. One thing I would say is that is true for free glutamates and aspartates, but generally speaking, if you have, in my experience at least, if you have a negatively charged residue which is involved in a salt bridge, it tends to be much better preserved for some reason that I'm not entirely sure of. But yeah, you do have much weaker density for glutamates and aspartates. They are still there quite often, but you have to take the threshold a lot down. Exactly, and you gotta sort of calibrate your expectations of what a residue is going to look like for Cryo-EM versus crystallography in that respect. So as I mentioned before, you're going to encounter situations where there is no unique placement of the sequence in the map. That is where there's unavoidable ambiguity. There's no real clear consensus on what to do in those situations. And when I say like unavoidable ambiguity, I'm talking again about like a combination between the information you have from the map and the information you have from your sequence. If you can place a domain but you can't see the side chains, that's fine. But if you're trying to build something de novo, let's say you're at 4.5 angstroms and you have a repetitive sequence and you just can't do it, you shouldn't, in my opinion, try to assign sequence if you're not sure, because that's like actively misleading. If you don't know, I would tend to err on the side of assigning the unknown residues with the UNK code, indicating they're an unknown amino acid. So they're modeled as alanine, they're modeled as polyalanine, but they're labeled to specify the fact that there is an ambiguity in sequence determination in that location. And the other ambiguity issue to think about is sidechain placement. So if you can't see the sidechain, but you know the sequence register is correct, do you place the sidechain or not? And there's sort of two main camps in the community there. One of which is just trim back all the side chains to fit the density. So that is, if it's a phenylalanine but you don't see the phenylalanine ring, trim it back to an alanine. And the other camp is place all of the side chains because we know that they're there, and then take care of annotating the occupancy of them either by refining a B factor or by manually assigning zero occupancy to the ones where we can't see any density. In some sense, the trimming approach is often considered more conservative. The one caveat to that is that it can kind of hide errors in your model because it doesn't take account of packing. If you have a sidechain that you don't see that's in the center of your model and it's interacting with another sidechain that you don't see, it's probably better to place those sidechains so that you can at least see that the model is not right? You allow them to clash, and you can see it provides additional constraints on your model building. But either way is fine. I would just say that be consistent and preferably specifically outline what approach you took when you're writing out that. So just some examples. So prior knowledge can come in many forms, and you should use any and all of those to guide model building. So this is a specific example. When we were building the initial structure of the ryanodine receptor, we were at 4.8 angstroms in that case, and at that resolution it was hard to. So this is a tetrameric ion channel, and so what we were looking at was a map like this, and just starting from that, it's hard to unambiguously know your protomer boundaries. And in this case, we were really lucky because if you have a look, we had one particular 3D class where an entire subunit or the entire cytoplasmic region of a subunit was missing. So that allowed us to guess at what the protomer boundaries would be, because it's more likely that an entire single protomer dissociated than some combination of two adjacent protomers, or at least that was the idea. And in other cases, you could also use cross-linking data combined with mass spectrometry, or negative stain of different complexes with and without a particular subunit. Any of these can be used to inform your model building approach. And also on this structure, if you're looking at ligands, I would advocate the use of difference maps, specifically locally aligned difference maps between different states where you have ligands present or absent, in order to confirm that you have the ligand that you think you have bound in the structure. In this case, we had 3 different ligands, calcium, caffeine, and ATP, and we were able to identify which is which by doing pairwise difference maps. So basically, if you do a difference map between a state where you have all 3 ligands and one where you have none, you see 3 blobs. If you do a difference map with one where you just have calcium versus nothing, you see only one strong difference density on the calcium. And if you do one where you have ATP and caffeine minus nothing, you just see that. You don't see any density on the calcium. So that. And when I say locally aligned difference map, what I'm referring to is basically you can do this in Chimera. You take a sphere of density around your ligand binding site of interest, extract it out from both structures, align them, and then subtract those. And the reason for doing that is to decouple any changes in the orientation of the domain that binds the ligand from that process. Because otherwise, if you just calculate a straight difference map, it'll look a bit messy, whereas that will give you, hopefully, better results. And yeah. This is just an example of you can get really nice difference density even at fairly moderate resolution in Cryo-EM. All right. So don't really need to go over this too much. This is just a general introduction to the Ramachandran plot, which I'm assuming you're all familiar with. But basically it describes the backbone angles, backbone dihedrals, that are allowable based on protein geometry. So which ones don't cause your adjacent residues to clash in a non-physical manner. And you can see that there are 2 main clusters in the Ramachandran plot, or 3 if you count the one over here. But you've got your alpha helical residues, your beta sheet residues, and then your left-handed alpha helices, and they contain a few loops in there as well. This is the Ramachandran plot for the general case. There are more specific cases for unusual things like prolines, glycines, residues before prolines, etc. That vary slightly. Okay, so one thing that I wanted to point out as well, or a consideration, is that not every alpha helix, or rather not every helix you encounter will be an alpha helix. There are other types of alpha helices which are populated in the PDB. And particularly if you work on membrane proteins, this is something to keep in mind, because although, with soluble proteins, alpha helices are by far predominant, in membrane proteins, there's a not insignificant fraction of 310 helices, including some fairly important ones, like, for example, the voltage sensor of a potassium channel. The S4 helix, which senses voltage, is typically a 310 helix. And at low resolution, that can be really tricky. The way that I would distinguish it at low resolution is that 310 helices are skinnier, basically, and have a triangular cross-section compared to an alpha helix. And it's something to think about just if you're trying to assign a helix and it's not making sense, It might be because it's a 310 helix or a pi helix, which has a larger number of residues per turn. That's less common as an actual long helix, but it's not uncommon to have a single turn of pi helix in the middle of an alpha helix. That quite commonly happens in ion channels. So it's something, again, to think about when you're interpreting your density. Something else, yeah, so this is something that can, unappreciated 310 helices can very easily lead to register errors, so they're a thing that you want to pay attention to for that. General introduction to beta sheets. Can be parallel or anti-parallel. The twist of the beta sheet varies, which makes them harder to build automatically and leads to more diversity between different beta sheet structures. And they're much harder to build at lower resolution. So where you can place a helix fairly reliably at like 7 angstroms, you really need to be at 4.5 before you can properly place a single beta strand. All right. So just thinking about some EM specific considerations for model building. So one of the disadvantages that we have is we don't have any sequence markers. So there's no equivalent of selenomethionine. There's no anomalous diffraction equivalent for EM. And this means that basically what you see is what you get, and everything that you're doing is guided from a single map, more or less, unless you make a mutation and calculate a difference map based on that or whatnot. But the other EM specific consideration is that you often have a substantial variation in local resolution, which is not something that you have in crystallography typically. And this means that different levels of modeling will often be required in different regions of the same structure. So you might have one region where you can just place a domain. You can if you filter your map to 7 angstroms, you can see it clearly, but you can't build an atomic model de novo into it. Whereas another region of the same map might be at 3 angstroms. That's not uncommon at all. And the other point I wanted to make is that resolution ranges that are very uncommon in crystallography are still quite common in EM. Because in crystallography, maps between 4 and 6 angstroms are kind of hard to obtain experimentally. It's unusual that you would get good phases to 5 angstroms and not be able to extend them further. In Cryo-EM, you get that all the time. And that leads to the case where you have to make a lot more difficult decisions when you're modeling in EM than you do in crystallography. Because you have the map to start with, you get that for free, and you've got to decide what information you can actually derive from it combined with your prior knowledge. And you'll also often have more than one map, like, for example, a bunch of different focused refinements that you will want to either model separately or to combine the maps and model one overall combined map. If you're going to take that approach, I would recommend the approach that I've found really useful for that is if you have separate focused refinements and you want to combine them into a single map, I would recommend the approach of taking the maximum value at each voxel between the different maps that you want to combine, as opposed to just direct averaging or masking and adding them together or whatnot. So that is, align the maps together that you want to combine, and then take the maximum value at each voxel. And you can do that in Chimera using the VOP maximum command, which I've put on the slide in case you need it. This seems to work because it gives you a fairly seamless combination between maps, because rather than just averaging two maps, one of which is good and the other which is poor in that region, you're taking just the density corresponding to the good one. All right. So where to start with building an initial model? We've already more or less covered this, but basically there are a couple of different situations. If you have a crystal structure or a homology model of a region of the sequence, that is always the best place to start. And I would start by trying to place that in real space using Chimera and then extending out from there. And if you have really good resolution, then by all means try auto building with Phoenix and see whether you get anything that looks sensible. The auto building routine in Phoenix has got a lot better recently, like in the last month or so. So it's well worth trying again if you haven't tried it for a while. And otherwise, if you don't have any of that, you need to take the approach which I mentioned. So identifying structurally distinctive motifs in the sequence, and then many of them in diverse parts of the sequence, and trying to locate them in the map, extend them, and stitch them together. Yeah. So here's a specific example. So this is a map of the ryanodine receptor. We had a crystal structure of the N-terminal domain that was previously solved, which you can see here in red. And we want to place it in the model. We can manually place it in the approximate location just by eye. And then we want to determine the orientation of the model in the map. So you can do that by what's essentially like global real space molecular replacement approach, where you can use this command fitmap with searching however many candidate orientations you want. Usually 100 orientations in a radius of 5 angstroms is where I'd start. Increase it if need be, but usually that does the job if you're roughly in the right location. And what you hope is that you will have one candidate orientation that is clearly separated from the others, that has better correlation coefficient with the map, and that should be clearly visually distinguishable as being the correct situation. Yeah, which you can see is the case here. One thing to be aware of here, when you're starting a project, if you have a crystal structure of a particular region of your protein, I would always use that to calibrate your pixel size, because the nominal pixel size that you're using, actually at SEMC it's pretty well calibrated, but depending where you're collecting your data, it may not be really well calibrated, and the only way to do it precisely is using the correlation of a crystal structure with a map, either your map or a map that was collected under the same conditions on the microscope. Usually once it's calibrated in that manner, it's fairly stable at the same mag on the same scope. And so this is an example here where there was a map that was deposited in EMDB where it had a nominal pixel size of 1.34 angstroms, and if you use the crystal structure that I'm referring to here and calculate real space correlation values at different nominal pixel sizes, you can see that there's a very clear optimum that's quite a ways away from that. Like 0.04 angstroms is enough of a difference that if you're comparing, it's fine on an individual level, but it causes problems if you're trying to compare like structures from different labs. Because if they're 2% off, that's on a 400 angstrom wide structure, that's more than you will see between many conformations of a protein. So it's something to pay really close attention to. All right. Okay, so once you've finished what you're doing in Chimera, you'll probably want to move to COOT. This is the software that I think is pretty much 100% dominant now for manual model building. There might still be a few people using O, but it's more or less mostly COOT now. And it's really simple and I think very intuitive interface for building models into density maps. You can run it on a run-of-the-mill laptop. And the nice thing about it is it has an extensive API which you can use to either roll your own functions or take other ones that people have created. One thing, if you've been using COOT for a while, I would suggest trying the latest nightly of the new version of COOT, which as far as I'm aware, at least for Mac, is currently only available with CCP-EM. So if you download the nightly release of CCP-EM, it has a precompiled version of the new version of COOT, which has some new fancy features for real space refinement into EM maps, building glycan trees, a number of different things that are quite helpful. I won't go into that too much. So as I mentioned, it's got an extensive API. I've written a bunch of scripts for it that you can download in a package called COOT Trimmings. If you have one of these scripts, basically the way to run it is you just copy it to a hidden directory in your home directory called COOT Preferences, and any Python file or scheme, which is a different language, but Python is mostly the one that people use these days. Any file that's put in that directory will be executed when you start up COOT. And so that allows you to define new functions for using the program, and also define custom key bindings, which I would highly recommend because it makes it a lot faster to use the program. And just to give you an example of what this kind of looks like when you're writing the function. So this is a little function that I wrote just to make mutating residues faster in COOT. So instead of having to click something and then click another thing, and then click another thing, it'll just bring up a little text box, you enter one letter, you press enter, and it'll mutate the residue. And that's really easy to do because there's a ton of different predefined functions in the COOT API that you can just plug into a very simple function and go. So and there's a lot of code that's already written that you can just copy, edit, and go. And so this is really useful. You can take whatever people have written, like things that I've done or things that other people have done, but it's also really useful if you have a complicated protein that you want to repeatedly do things to. So if you want to, for example, do a domain-wise rigid body refinement into a set of different conformations of your EM map, for example, you can easily script that in a little bit of Python code in COOT, and it's really quite straightforward to do using exactly this kind of approach. And all of those functions more or less are documented in the COOT, well, I shouldn't say all, but there are a lot of those functions that are documented in the COOT user manual, which you can then just use directly. It's pretty straightforward. Okay, so some things, so there are tons of key bindings. It's really easy to define your own custom keys. And then you can get to those from the settings, and it'll tell you where all the standard key bindings are and where all the user-defined key bindings are. So in my COOT Trimmings package, it'll give you a new custom menu, which should show up and has a bunch of different tools that you can use for both visualizing your protein, so like custom coloring by hydrophobicity, coloring by finding particular sequences, a lot of different things. If you want to use it, give it a go. You can use on the fly B-factor sharpening, which is really nice if you have your map as an MTZ file. You can't do it with an MRC, but if you convert your map to MTZ, then you can use that, and that's nice for just trying to identify, what the optimal sharpening factor is for your map, particularly because it may be different in different regions of the map. And you can also change the resolution limit of the map if you just want a low-pass filter on the fly, like filter it to 4.5 angstroms just to see a particular disordered region or something. That's the thing that you can do. Honestly, the B-factor sharpening is probably the more useful approach for the same thing, but it's always good to have options. COOT also has semi-automated helix placement in built, which is probably the place that I would start for assigning secondary structure. Helices are the easiest and the best place to start, so use them if you got them. I would suggest binding that to a key. In COOT trimmings, I have it bound to H, so you can just navigate to the center of a helix, press H, helix will appear, and then you can try to grow that helix or by a certain number of residues if you can see that COOT didn't quite make one that's long enough, or trim it if need be, and then refine it into the map using real space refinement. So you might want to grow it by, let's say, 6 residues. It'll grow down there. Then you can refine the whole thing into the map, drag it around to fit the density, and then the next task is to assign the sequence. To do this, my approach is I'll usually just renumber it based on the sequence and then mutate it based on that numbering. So I have some tools that just make it a little easier to renumber in COOT. So you can just center on a residue and renumber based on what you want that residue to be, as opposed to calculating an offset of some sort. And then you can mutate that region to the sequence by just pasting in your sequence into the text box. It'll strip out all the formatting characters or anything that's not an amino acid automatically. And then you can check whether this is a way to test different sequence registers. So you can try this candidate numbering for that amino acid, try another one, mutate the whole sequence, and see if it's consistent with the density. Right, so I'm sort of getting close to the end. I don't have that much more, but one thing I wanted to briefly cover was just to think about the types of errors that you can have in macromolecular models. So you can have errors of identity or directionality, which are most common at very low resolution, so sub sort of 4.5 angstroms. So that means putting a domain in the wrong place or tracing a chain backwards. Those are both things that happen and have happened quite commonly in low resolution EM maps. Probably, and then at very high resolution, you can have errors of rotamer placement or backbone torsion, so having a carbonyl that's the wrong way around, or ligand identification, or the pose of the ligand. But probably the ones that are most common in the cases that you'll encounter are errors in topology and connectivity and register. So is this helix connected to that helix, and what is the exact local sequence register in this particular region of my protein? And register is by far the most common one where you will encounter errors, either in models that you're building or in models that you download from the PDB. And I would always encourage, if you're, as a consumer of macromolecular models, I would always look at the model in conjunction with the raw data. Always look at the model with the map, because you'll often be surprised how marginal the data is that the model is built on, and it's good to know what you're dealing with. So this is an example of a register error where, if you're building and, you might get it wrong, I'm not sure. Yeah, so that is not a real example. I made that one up. Okay. Just to be clear. But just to illustrate the point. Although there are...
Oh, yeah.
There's one particular structure I know of where an entire several thousand residue region was traced backwards. But I'm not going to name names. Yeah, so just thinking about this stuff, so you should have a strategy for trying to identify and correct these errors as you go. So I would strongly encourage you to analyze the structure as you build it. Don't wait until the end and then analyze it. Analyze it at every intermediate stage of model building in terms of mapping conservation on the structure, mapping hydrophobicity and polar and non-polar residues on the structure. Try to get a sanity check on whether everything's making sense, or whether there are regions that, even apart from the density, you can see don't make sense. if you've got a ligand binding site and mapping conservation on the structure doesn't automatically highlight that, then you might want to think about whether there are some errors there. Likewise, if you've got a membrane protein and you're finding that you don't have a classic belt of hydrophobic residues around where you believe the membrane is, that's another warning sign that maybe some of those are not quite right yet. And as I said, use like secondary structure predictions and disorder predictions, not as a validation per se, but as just a check to make sure that everything's making sense. EM Ringer is really helpful at slightly higher resolution to identify errors in backbone and rotamer geometry. And just in general, look at everything. If you're building a structure, you should be looking at every residue. Even if you've placed a pre-built crystal structure, you want to be going through residue by residue, making sure that everything you have manually inspected, because that is by far the best way to identify errors in your model. You can run as many automated tools as you like, but given that most of the time when you're running refinements, it will be optimizing the geometry, it'll usually hide a lot of those errors from your site in any automated output. And as I mentioned, sometimes you just can't tell the right answer, and don't be afraid to say that and to specify ambiguity. Oh yeah, half-map FACs. So this is a very common thing that one does in order to analyze whether you have any overfitting. It is useful for that purpose. It's not useful for any other purpose. It only tells you something about whether you have any overfitting, like globally, and what the global map to model fit is. It won't tell you if the sequence register is wrong locally. Like, that's not a thing that you can derive from the FSC. It's only a measure of global agreement. All right. Right, so I don't really want to go through this too much, but this is just a basic. So this was the case where we had a 4.8 angstrom map. We started off with that map, a crystal structure of the first 500 amino acids, and a couple of domains. We could already see from the density where the transmembrane region was, and we knew from analysis of the sequence that the transmembrane helices were right at the end of the sequence. So basically we were anchored right at the end of the sequence, we were anchored at the start, and we had to build everything in the middle. That allowed us to build a polyalanine trace that we could then use to identify domains that were known, or to putatively identify domains that were known based on homology, and also some that weren't. So like all of this stuff around here, this was all the gaps in that initial map of conserved domains that I showed you, and it turned out that basically all of the gaps were alpha solenoids, so helices of helices, repeats of one helix after a helix of helices. And as I mentioned, a fortuitous 3D class with a disordered protomer allowed us to determine the protomer boundaries, which you can see here. Now, even...
Quick question.
Yeah, please.
On that slide before,
yeah.
when you're looking at that bottom right hand, like the bottom side of your...
Why is there all the density there? So that's detergent. So for any membrane protein, you're usually going to be extracting it out in some surfactant.
Sure. I was just wondering why there was no
model there.
Yeah, that's it.
Yeah, so that's the detergent belt. If you had a nanodisk, it would extend out a bit further. So yeah, this allowed us to think we identified all the domains. One note of caution, like even in that case, we were at 4.8 angstroms and we got these guys wrong. So we had there are 3 repeats of this one domain, the SPRI domain. And we had 1 and 3 swapped around, which a competitor of ours got right at the time, and we since corrected in a subsequent publication. And even going back to it, I still can't see, I don't think we could have known based on the density at the time. But it's, always be skeptical of your own assignments. And we now have much improved resolution. We have better resolution than this now, actually, but at improved resolution, you can identify things like calcium ions. You can clearly see, and so this is an example where you can clearly see, like, this glutamate is pretty defined because it's binding to this calcium. Like when you have negatively charged residues that are either interacting with ions or with positively charged residues, they tend to remain pretty well defined in the density, in my experience. One thing I'd point out here is that even at our best resolution at the time, we could not assign more than 1,000 residues, so they were all polyalanine, no sequence assignment. In that case, I would assign them as such, but I would also go back to your data set and see whether there's anything that you can do to get a better map locally that will allow you to get improved sequence assignment. In this case, we had the idea that that might be because of basically continuous conformational heterogeneity in the structure. So this is looking at several different classes of the same data set, just interpolating between the maps. And you can see that the region which was poorer resolution is also one of the regions that's moving around a lot. So we had the hypothesis that if we performed a master refinement of that, we could get better resolution. Because this is a tetrameric protein, we needed to perform symmetry expansion first. After you do that, you can then mask one asymmetric region, but enclose all of the asymmetric units effectively, and then perform a refinement in C1 with local angular searches. And when you do that, you do get greatly improved local resolution, which you can see here. And so now a lot of the regions which were previously poor are much better, and everything outside the mask is washed out. And so that allows us to go from things like this, where you can maybe see this was one region where we had a crystal structure, so we could place it, but you really can't see much. And then after refinement, it's a lot better. And so this allows sequence assignment of regions that we didn't previously have connectivity or side chain definition. And that's all I've got. I'm sorry, that was only a little over 1 hour, but I'm happy to take as many questions or just discussion as you guys feel.
Get us started. So I've done no model building, so this is always a good lecture to learn from. If I'm just lazy, and
being lazy is good.
Yes.
Yeah.
If I'm just lazy or I just don't know,
Yeah.
what's the lowest resolution that I can start automated map building?
Oh.
Yeah.
I mean, you could try at 3.5, but honestly, I still wouldn't advise it anyway.
Okay, but like even as a starting point, if I wanted to.
As a starting point, it's fine, but it depends largely on what other information you have, right? Like in this particular case, are there any other proteins that have domains which are solved? Is there any information that it's always going to be a much bigger shortcut. Like the lazy approach is to have a look at every database that you can and see if there is a structural homologue of a particular region that you can place and then extend. That will be much faster than any automated building procedure as of right now. Because what you'll get out of an automated building procedure is a bunch of fragments of different lengths, some longer, some shorter, and you will then need to manually identify which of those agree with the density, which don't, and connect them. Certainly at 3.5 you could give that a shot, but I would still recommend use every other bit of information that you have before pressing the go button on an automated building procedure.
And what's the time scale for something like that?
That depends.
Like a 200 kilodalton.
Oh, you mean for the automated building?
Yeah. Like I've never done it before, so.
I mean, I don't know, to be honest. I did try the other day using Phoenix Map to Model on a 300K protein, and it chugged for a few hours and then I killed it.
Okay.
But that's going to vary a lot depending on your particular situation. But I think, I'm definitely biased, but I would say that learning manual model building will stand you in good stead also for interpreting the results of any automated output, because the model is really only going to be as good as the interpretation of whoever's looking at it.
In my experience, the automated model building, it doesn't really work if you have like disordered regions in your domain, for example.
Yeah.
It doesn't work at all.
Yeah.
It gives you a bunch of crap.
Okay.
Yeah, I haven't had good experiences with it yet. As I say, Phoenix has recently improved their algorithms in the very latest version of Phoenix, and it's supposed to be a lot better, but I haven't had, and a lot faster. in Phoenix 1.15, but I haven't had the chance to test that extensively yet.
Have you used the sequence from map for de novo building in Phoenix?
Yeah, I've tried. As I say, for my cases, I haven't had great success with that, but it'll vary on each individual case. But looking at the statistics for that, even in the best case, you're getting out a model which still needs quite a lot of editing and completion.
So any other tools that you would recommend for de novo building, like Rosetta or
Rosetta,
try that?
I've tried Rosetta. Rosetta is great. It's got a fairly steep learning curve for getting used to, but in particular situations, it can be very useful. It is also quite heavy on the computation side. So if you can build your own model first and then extend it or rebuild it using Rosetta, that's probably at this stage still a more productive way to go, I think, if you can, than trying to build completely de novo using Rosetta at this point. But I'm not the Rosetta expert.
What about, do you have like a recommendation for like your favorite tutorial that shows you how to do model building? Like any resource, like for someone who's never done it before.
I mean, so I don't know really. There aren't any really good ones for manual model building.
Okay.
You just gotta, I mean,
do
there,
it.
there may be. I don't really know any though. I think the best way is just to sit down,
Experimental data sets.
sit down with COOT and a map and a model. Doesn't
COOT
have to
has
be
a
yours.
tutorial which
COOT does
I
have a
found.
tutorial, but it's like very high resolution.
Right.
It's at which point that's useful for understanding the basics of how to use the program, but it doesn't tell you how to use the program
For your
with your
data.
shitty data, like which is what you really want to know, right?
Sure. Okay.
Yeah.
What about incorporating mass spec cross-linking data? Is
I-
that just a sanity check, or can you
I
actually
think you want to be pretty careful there to make sure that how much you can trust the data that you're looking at. With mass spec cross-linking data, quite often in my experience, you want to be careful that the cross-links that you're looking at are meaningful. Because if you have a protein because we had this experience where another group was building their antigen receptor and they used cross-linking data to inform their model building. But the antigen receptor has a giant number of very long disordered regions. And most of the cross-links that they identified and used to build the model were cross-links between that disordered region and other regions that were either disordered or not, but didn't really matter. They weren't terribly informative. And so they can be, I would use them more as a validation than anything else if you're going to use it, or be careful with how you're doing the cross-linking, because they can be actively misleading in that case. Like if you're looking at it, because you don't know to start with that region is disordered necessarily, so if you're using that to guide your model building, it can be not helpful sometimes. Probably sticking to fairly short cross-linkers and maybe limited proteolysis of your protein or something would be a way to go. Like you want cross-linkers that are only cross-linking things that are actually structured, because otherwise you're going to get out a lot of data that's difficult to interpret. Some people have used the evolutionary couplings really well in that, using that same kind of approach though. So there's Gremlin and EVFold, two servers that you can use for that, for identifying evolutionary couplings between different positions in your sequence. And that can be useful both as a check and to try to identify whether like a particular candidate orientation of a binding partner in a complex makes sense, for example.
Any more questions? All right, so let's move on. So I think this is the last lecture in the EM course. Ed's going to give a wrap up on Wednesday, and then we're done. So thank you everyone for your participation. Thank you to the speakers especially. Yeah.
Can we get a slide something?
Yeah, of course.
Yeah, the slides are
That'll
gonna, if you
be-
would send it to me,
I'll send them
PDF
to-
format would be the
yeah.
best, and
Yeah.
then I will upload it to our website, and if you go to semc.nysbc.org, we have all these upcoming events, and the EM winter course is one of them, and all of the different lectures from, like, every single lecture is on their PDF format, so you can grab their slides from there, including the videos, which are also on YouTube, so.
Thanks.